All right, it's my honor and pleasure to introduce Dr. Mary Klotman today, who's going to give us the second talk about HIV and the kidney, an organ near and dear to my heart as a nephrologist. Dr. Klotman earned her undergrad medical degree, internal medicine residency, and fellowship, all at Duke, so the 4 Duke, as you said. She was an assistant professor at Duke for a short period of time before going to the NIH, where she was a member of the Public Health Service. In 1994, Dr. Klotman joined the faculty at Mount Sinai School of Medicine, and she ascended the ranks to become a tenured professor in medicine and microbiology, and held the Irene and Dr. Arthur Fishberg Chair in Infectious Diseases. She also served as the Division Chief of Infectious Diseases for 13 years and co-director of the Mount Sinai Global Health and Emerging Pathogens Institute. And then I guess she went back to her first love, which is Duke, and became again chair of the Department of Medicine there, and in 2017 became Dean of the Duke University School of Medicine and Vice Chancellor for Health Affairs. She is an accomplished clinician and scientist. There's more in her CV than I can go through in the short time that I have to introduce her, but her research interests have focused on the pathogenesis of HIV, and among her many important contributions, Dr. Klotman and her team demonstrated that HIV resides in and evolves separately in kidney cells, the so-called renal reservoir, which was a new nomenclature at the time. And as I shared with Dr. Klotman, I remember preparing my talk, my senior talk as a fellow on HIV, and that was kind of the highlight of the whole talk. So more recently, her group has been defining the role of integrase defective lentiviral vectors for the delivery of an HIV vaccine. So it is my pleasure and honor to have her talk today to us. Thank you.
Good morning, and thank you for inviting me. I can say I came because I thought it was a great topic and an exciting opportunity, but it also is a nice break from being a dean. So it is a pleasure to be here. And the topic is near and dear to my heart. In fact, it's something I have studied my whole career. You know, occasionally I've participated in those existential discussions about the death of physician scientists, and I agree wholeheartedly that it's quite the opposite. I think it's incredibly exciting, the breadth of careers now that you have to choose from in academic medicine, all the way from traditional laboratory-based science to implementation science to data science. My existential moments are when people say academic medical centers are dead. You know, it makes me pause a bit, and I love to use the HIV story as exactly why we need academic medical centers. There's no question in my mind that this very, very challenging and really incredibly, I think, incredibly devastating disease would have not been solved if it weren't for the incredible collaboration between academic health systems willing to invest the money, resources, and invest in the education and training of physicians, funded by the NIH in partnerships always with pharmaceutical industry. This was a remarkable collaboration, and I think we need to continue to be the drivers of this kind of incredible story. So I may be self-serving, but I think the HIV story is one of the reasons to have academic medical centers. So anyway, I have been in this story from the beginning. I too go back to when HIV first hit the wards and saw some of the first patients at Duke when I was a resident and fellow. And my career really has followed the epidemic, particularly as it relates to the kidney. So today I'm going to go over the history of HIV, and most of it is history. Sometimes we get nostalgic talking about history, but this is claiming victory. Really talk about how we worked on the molecular pathogenesis of HIV and the changing spectrum of the disease, particularly with the onset of antiretroviral therapy. What we know now about a different spectrum of disease with the interaction of HIV on renal function, implications for cure initiatives, which I touched on a little bit in my question, and then a very interesting, I think exciting turn of events where we're doing HIV positive to HIV positive organ transplantation, but how we can use that to even further understand what the implications of HIV are in some of these organs that are being transplanted. So with that, I always begin talks, and I used to make sure every grand round began with a case. And I'm going to go over two cases, one of which I published, one of which was one that I saw. The first is a case in 2001, which really reflects, I think, what HIV nephropathy looked like for many years. This was a 30-year-old black male who presented to our emergency room at Mount Sinai with fatigue, weight loss, night sweats, rash, impressive adenopathy. Now, despite the fact it was 2001, where I would hope that everybody was very much aware of HIV, particularly in Manhattan, Upper East Side, we were at one of the epicenters, he was admitted with the presumptive diagnosis of Hodgkin's. I saw him that evening. He was very sick. Take a history, as good ID folks do, a sexual history. He had multiple sexual partners. In fact, he was getting tested pretty regularly, but he had recently seroconverted, unbeknownst to anybody, and had a very high viral load. Long story short, he had massive proteinuria. He was in really accelerated renal failure. He had a biopsy, and I'll go over this as a classic finding for HIV nephropathy. Initiated dialysis. Now, we're very fortunate that we had a clinical protocol, IRB approved, to do biopsying both before and after initiating therapy, and that has really led to some of our major insights, a case of one. Initiated dialysis, and Art came off dialysis within 2 months. His renal function recovered, and the rest is a little bit of history for him. Fast forward 2013, this was a case that one of my nephrology colleagues, Christina Wyatt, saw, a 30-year-old Hispanic black male. He had non-nephrotic proteinuria. He was on antiretroviral therapy, had most of the time non-detectable viral loads, and a robust CD4 count. And he had a different pathology, so he didn't have this collapsing glomerulus, but he did have some interstitial inflammation, some sclerosis and fibrosis, and he had this microcystic tubular dilatation, which is one of the hallmarks of HIV. So the question has been, and still is that HIV? I would say yes. So what was HIV? Well, I say was, but we occasionally see cases, and it always surprises the house staff because, again, it's in the history books in many, many centers. But the first reported case was in New England Journal in 1984, where at the time 10% of patients at a single New York institution, this was NYU, presented with this picture of massive proteinuria, rapidly progressive renal failure, usually seen in late stage AIDS, although I reported one of the first cases in early HIV, largely seen in those of African descent, and the genetic history has been absolutely fascinating in this disease and opened up a whole broad area of research. And it was for many, many years the most common cause of chronic renal failure in HIV-positive, seropositive black patients, particularly in urban settings. Clearly, antiretroviral therapy has changed the disease phenotype, but I would argue it has not gone away. So what were the pathologic findings? Interestingly, large kidneys on ultrasound, which was one of the hallmarks, not amyloid. The other cause is HIVAM. And it had a collapsing FSGS with tubular microcystic dilatation, massive leukocyte infiltration, sometimes it almost looked like a proliferative disease, interstitial fibrosis, and podocyte proliferation. This has been the focus of over a 20-year program project grant that I have had the pleasure of participating in, where we really used both animal models, cell culture, and human biopsies to work out the pathogenesis, and I'll just go through that briefly. We were very fortunate when I was at the NIH, they were creating some of the first transgenic models for HIV. Just a little side story, you couldn't put the whole genome into mice. There was real, I think, reasonable concern that it might replicate, and you could imagine a mouse escaping in the middle of DC. So in fact, the first transgenic mice they held in facilities that had chloride moats around them just in case the mice started to dive out. So there was a lot of unknown. We now know the virus cannot replicate in mice, but nevertheless, you could only work with a partially deleted genome. In this case, the GAG pole of the genome was deleted, but the construct had all the other, what we were calling accessory genes of HIV, because we weren't really sure why they were there, like NEF, VPR, VPU. And lo and behold, the mouse lines had a pathologic kidney disease. In fact, that's the only disease they got because it does not replicate in T cells in mice. But this looked like HIVAN, and that was the aha moment for me. My husband is a nephrologist. We tell this story with a little bit of different history. I say, he was in another lab, came to me and he said, I was looking at these slides today from a transgenic mouse, and it kind of looks like, the human HIVAN. And I thought, oh my God, this is the first small animal model for HIV. He doesn't tell it the same way. But anyway, we have used this model to really exploit and figure out the molecular pathogenesis. So as you can see side by side, the biopsies look very similar between the transgenic line, TG26, and a human biopsy, and a whole series of experiments, cross-transplantation studies, which are really cool, going from a transgenic mouse into a non-transgenic mouse, making transgenic mouse lines, deleting each one of the genes. You can imagine all the different things you can do if you have a really great model for a disease. But bottom line, it showed that expression of HIV as a transgene did recreate the whole clinical and pathologic phenotype. Many models have confirmed that, including rat models, that the renal disease was dependent on expression of the transgene in renal cells. And that was, very sort of unusual finding, because it's not, if you did a cross-transplantation model, that is, you took a normal kidney and put it in the background of a transgenic HIV mouse, did not get the disease. So none of the circulating cytokines, chemokines, etc. contributed to the disease. But if you did the opposite, which is take a transgenic kidney and put it in a normal mouse, it got the disease. So that made us go back to human biopsies and say the virus has got to be there, even though it had not been found, and I'll go through that. But again, we used it to work out the direct role of accessory genes like NEF and VPR in disrupting podocytes and renal epithelial cells, and that has been a lot of fun, just to kind of be able to really go down to the molecular level and map out what this virus does in a cell that ultimately affects organ function. But I'm not going to focus on that. So what we did do, though, at this, when we really got the results of the reciprocal transplantation studies, is go back and look very carefully in biopsies from humans. Now that meant literally going into the biopsy suite with the prep in hand, because you get rapid degradation of viral RNA. And just by doing that, and then doing in situ hybridization, we were able to show that the majority of biopsies actually had both viral RNA and DNA in a really interesting distribution of renal epithelial cells and podocytes. And this was, interestingly, in our first series, we didn't pay too much attention to what the pathology of the biopsy was, so we kind of looked at the whole series of biopsies we had. Some of them did not have HIVAN. So that opened up another series of questions, is the virus has to be there, but it's not sufficient to create the disease. And in fact, now we know the other piece is the genetic predisposition to renal disease. That is the APOL1 story, which we did not work on, but I think it's just an amazing breakthrough in understanding the contribution of genetics to disease, particularly kidney disease. But anyway, we continued to work on what the virus was doing in these kidney cells. By the way, renal epithelial cells do not have your classic receptor or co-receptor, so a lot of skeptics were saying, it doesn't have the right molecules on the surface. How does the virus get in? And I'm going to go through that story as well. But what we were able to do is really start to address this issue. Is the kidney a unique compartment? And I'll get down to reservoir, but is it a unique compartment for HIV? In other words, does the virus evolve differently in the kidney compared to the evolution of the virus in the blood? And what is really a great tool in HIV research is the genetic sequence of each virus can be used like a fingerprint, because it evolves so fast that you can really tell relatedness based on the exact sequence of the virus. And so you can build these viral DNA trees looking at sequence, and what you're seeing here is laser dissected and captured DNA from renal epithelial cells, and sequencing that envelope DNA within the epithelial cell and comparing it to the sequence of viral DNA in peripheral blood cells and plasma. And just by looking at the color coding, the sequences in the boxed regions are kidney sequences. The sequences in the non-boxed are peripheral blood or plasma sequences. And basically, you can see the box sequences form their separate tree, suggesting or supporting the idea that the virus within the kidney is evolving under different environmental circumstances or pressures compared to virus in the blood. And therefore, the kidney is indeed a compartment. That doesn't mean it's a reservoir. But again, that was another surprising finding. Now, the one other area that we started to pursue very aggressively based on the finding viral DNA in the kidney was to ask, how does the virus get in these unique cells? And we were really impressed by the robust T cell infiltration in the kidney, and somebody not on therapy, they all have virus. So this is just looking at the HIV RNA in interstitial T cells, and that kind of led to coming up with a model that I'll show you about how the virus actually infects the renal epithelial cells. So the human biopsy studies that kind of complemented what we were doing in mice showed us that HIV does infect epithelial cells, that there's this robust interstitial inflammatory process with infected T cells. The other interesting finding I didn't go over too closely is that within a biopsy, you have one tubule that's all infected sitting next to another tubule that's not, really suggesting cell-to-cell spread once you have an infectious event occur. And as I said, because we found virus in some of the biopsies that were done because of diabetes or other causes of renal disease, we really concluded that the virus was required but not sufficient to cause HIVAM. So we did go after how the virus gets into epithelial cells, and we set up a co-culture system where we had labeled epithelial cells, labeled T cells, labeled virus, did a whole series of experiments, putting both primary as well as transformed renal epithelial cells in co-culture with T cells, and found that you could have massive transfer of the virus from a T cell to an epithelial cell only if they came in direct contact. And so that the infection required direct contact. And then we've modeled this out in a number of experiments and showed that you get virus integration in the epithelial cell, and once you have virus integration, that epithelial cell can make virus much more efficiently, spreading virus to a T cell, again with contact. So we have developed this whole model of an infected T cell coming in contact with an epithelial cell, and by the way, you have a lot of breakdown in the basement membrane in the biopsies of these patients, so it can be on either the basal surface or perhaps the apical surface. But once you have that contact, you have spread a virus into the epithelial cell. In some cells it gets integrated. Now, renal epithelial cells do not necessarily turn over. And so again, that observation started us to think down this pathway of, is this a reservoir, meaning a long-term site that can be maintained in a host and down the road actually lead to reactivation of replication-competent virus? And I'll go over where we are on that. But this is the model we've really worked on over the years. That is, in the absence of antiretroviral therapy, you have a tremendous amount of inflammation, including chemokines that bring T cells into the kidney, that in that setting, you do have spread of the virus by contact to epithelial cells. In the presence of antiretroviral therapy, a lot of that inflammation goes away. In the absence of a stressed kidney, those cells have integrated DNA and can have integrated DNA for the life of the cell, which can be the life of the host. But with stress, that cell could reactivate the virus, and in fact, really interesting study came out from a French group a couple of years ago where they were participating in an HIV transplant study, and in this case, it was negative kidneys into infected recipients. The recipients were completely suppressed, no plasma blips of HIV. They did a series of biopsies post-transplant, and the majority of the kidneys that were transplanted as negative became positive. The in situ hybridizations looked identical to what we had published 10 years before. In fact, I almost thought they were ours because the patterns were so distinct. But it raised the question, how did the virus get there again? I think it's an activated T cell, because the T cells we know we have a very robust reservoir of T cells harboring viral DNA. And so with any kind of an activation event, it's not hard to imagine, particularly an allo reaction, that you might be able to activate and transfer the virus. So another part of the story of HIVAN that is not our story, but it's just an amazing breakthrough. So we were doing these cross breeding of mice, and we actually showed that some mouse strains didn't get HIVAN despite having the transgene, couldn't figure it out, trying all these back crosses. It's very laborious. And meanwhile, there were several groups doing GWAS studies looking to see whether they could map what the genetic predisposition is to HIVAN, and sure enough, they did. This has been an amazing story with a lot still unanswered, but basically there's two APOL1 missense variants, G1 and G2, that are strongly associated with the risk of HIVAN. As I said, these are really important observations. 29-fold increased risk of HIVAN if individuals have these two alleles. And the alleles have an additive effect. Interestingly, when groups looked at other forms of renal disease that had known to have higher prevalence in those of African origin compared to Caucasians, sure enough, these variants predisposed to FSGS, hypertension, sickle cell disease, lupus nephropathy. So this is breakthrough. And I'd love to think that many of the observations in the HIV opened up other fields. So the question then became, well, why are these alleles so maintained throughout a population of individuals that had long ago migrated out of Africa, and they were maintained actually throughout the world? Well, it turns out that these variants also confer resistance to African sleeping sickness. And so they had a very positive protective effect, particularly in Africa, that maintained them at high rates within the population. So, fascinating story, but I don't have time to go through why APOL1 predisposes, and I would argue we don't know yet, but there's a lot of science going on there. But let's get back to what happened with antiretroviral therapy. So if you just look at epidemiology of end-stage renal disease in HIV-positive individuals, you see this very significant inflection point. It's a similar inflection point in time when mortality started to drop, and obviously that's when antiretroviral therapy really started to spread. But you also notice that in the black line, that although the end-stage renal disease in HIV-positive individuals started to go down, it hit a plateau, despite widespread distribution of antiretroviral therapy in this country. Africa, this is a different story, where you don't have, in some areas, widespread distribution, and you do have a lot of renal disease. But there is a plateau here, and I'll go through what I think that is. So there clearly was an inflection point where the manifestations of HIV relative to renal disease really changed. So if you look at a biopsy series at Mount Sinai, published by my longstanding colleague Christina Wyatt, you can see that somewhere around 2000, 2001, the percentage of biopsies with HIVAN, which was the predominant diagnosis, fell dramatically from over 60% being HIVAN to under 30% being HIVAN, and the rest being a mix of the other diseases that both HIV positive and HIV negative individuals get. Hopkins had a similar series showing that between 95 and 2004, HIVAN diagnoses by biopsy decreased from 80% to 20%, with other diseases like FSGS, interstitial nephritis, some from HIV meds, diabetes, hypertension, hepatitis C associated renal disease. They all started to actually go up as a percent. Now, one challenge with what's happened over the years is we have less opportunity to look at biopsies. So there's a huge selection factor here, because when HIVAN was so unique, obviously we wanted to biopsy, see what that is. I don't know about my colleagues at other institutions, but it's very hard to get nephrologists to do renal biopsies these days. But there are more subtle effects of HIV on kidney function. So this is a very large VA study with an aging cohort, simply looking over thousands of patients, relative cofactors for development of chronic renal disease. And if you look at the open boxes, in every case, age cohort, when you're comparing HIV positive to HIV negative, HIV positive increases the risk of chronic kidney disease by as much as 2 to 3-fold. Other things do as well: hepatitis C, which we know, hypertension that we know, diabetes that we know. By the way, HIV infected patients get all of those. So you start to have complex interactions between multiple cofactors contributing to CKD, but nevertheless leading to a higher risk for CKD in an HIV-treated population compared to a non-HIV population. And this is one of the smaller studies coming out of that cohort study showing the influence of the combination of HIV and diabetes. As you can see here, the combination of the two shows more progression to CKD compared to either HIV alone, diabetes alone, or having neither one. So we were able to model some of those interactions using that TG26 line that has the provirus and creating diabetic models. And again, showing that if you have the HIV transgene in a mouse model and you create diabetes, you have a much higher incidence of proteinuria and progression to renal failure. So there are clearly interactions between these other diseases like diabetes and HIV on the influence of kidney function, the influence on kidney function. Some of it definitely has to do with chronic inflammation, which was clear in these animal models where you had higher levels of inflammatory markers that correlated with the enhanced progression in diabetes and HIV. And that is turning out to be true in many of our chronic disease studies in humans. So about the mid-2005 to 2010 years, there were many large cohort studies, they all have great acronym names, that clearly showed that there are serious non-AIDS events occurring in suppressed HIV-positive populations. And these events had a lot to do with organ damage, including cardiovascular, liver, non-AIDS malignancies, and renal disease showed up in 3 of the studies as well, kind of associated with accelerated aging, but again, that's a catch-all word for a very complex interaction of inflammation. But it is really turning out to be the role of chronic inflammation. So, getting back to the first case that I studied, which really kind of raised another set of questions for us, that individual that we treated and were able to do a pre-treatment biopsy showing high VAN. Several months later, clinically doing much better, off dialysis, did another biopsy. A lot of the pathology resolved. Proteinuria went down to normal. Creatinine recovered. This is just showing some of the residual scarring during therapy. But we did do in situ hybridization, both pre-treatment and after what we thought was successful treatment. And what's most striking is, despite all the improvement clinically and the pathology, that the amount of nucleic acid never changed in the post-biopsy compared to the pre-biopsy. It makes sense. Our antiretrovirals have no effect on integrated viral DNA, no effect on transcription that would lead to viral RNA. So not surprising on the one hand, but pretty jarring on the other hand, because you see on therapy, that's a lot of viral nucleic acid. So that really led us to start focusing on this issue. What is the long-term consequence, be it thinking about cure or thinking about kidney function, of having integrated HIV DNA in patients over many, many years? And so that gets us into the cure science of HIV. So there are a lot of sticklers in the field that really try to standardize these definitions. So when we talk about a compartment, which I did talk about, that really is an anatomical site where virus can replicate with limited exchange of other sites. So you start to see an evolving set of genomes that look different. And I think that was obvious on our laser capture dissected study. We talk about latency, that's a non-productive state. virus is integrated, but you're not producing any virus. A reservoir is a cell that harbors replication-competent virus. So the vast majority of cells with nucleic acid, the cells that I showed you in the situ by DNA hybridization, may not have a full genome. In fact, we know they don't have a full genome. And so they can't produce replicating virus. And so at one level you say, well, who cares? There's a lot of junk DNA. But about 1 in 1,000 to 1 in 10,000 can produce replication-competent virus. And as we know in many of the studies where we've stopped antiretroviral therapy, it only takes a few cells to have replication-competent virus to have a robust rebound. So we have been really focusing on, well, what does it mean that renal epithelial cells have integrated provirus, particularly as we think of some of the cure strategies. None of them have shown dramatic success, but they're interesting. So one is the strategy where you activate virus in every cell that you can and massively kill cells that are infected under controlled circumstances, and that ideally you could do that in repeated cycles and eventually get rid of all the cells that have competent virus. Hasn't played out dramatically, but again, it makes you think, well, how do you activate virus in renal cells compared to peripheral blood cells? And that is an area of active study in my lab currently. But the other challenge to studying the kidney as a reservoir is you can't keep biopsying patients over time to see if the virus is evolving even under therapy. So what we started to do a couple of years ago was really refine our methods to look at virus in the urine, and sure enough, if you look hard enough, it's there. So here I'm just showing genetic trees. The triangles are genomes from urine, the red are genomes from blood, and as you see here in 3 of the 4 patients, the triangles are starting to form separate branches compared to peripheral blood genomes, supporting this idea that the GU tract is a unique compartment. Now, the GU tract is long, has a lot of compartments in it, and currently we're comparing what we're getting from the urine with what we're getting from seminal fluid to show that the urine actually, in many patients, reflects a compartment of virus that's different in men than seminal fluid, and obviously in women it is clearly a unique compartment. So this is becoming a tool to be able to study virus over time in what we think is the kidney reservoir reflected in the virus that we're getting from the urine. And sure enough, we are doing serial urine studies, and I don't want to go into it too much detail, but this is a patient that we've done collections over many weeks, and what we see is this dramatic separation of the virus in the urine compared to the virus in the blood. So more to come on understanding what happens in this compartment and reservoir over time, and then certainly what happens in some of the strategies for cure. So we're doing things like reactivating virus in culture, in epithelial cells, to see if the dynamics and kinetics look different or similar to peripheral blood. But there's another opportunity that has come our way. I think it's wonderful that with the HOPE Act that we are able to transplant HIV positive kidneys that would otherwise be discarded into HIV positive individuals. But that also provides us a unique opportunity to ask, okay, you've got genetic tags of virus, and you can actually follow the virus. And so we do have an active protocol to assess whether virus populations from positive donor kidney can be found in urine and blood specimens of the transplant. The short answer is yes. Now, do I think that's horrible? I have no idea whether that's bad. I am a strong believer that antiretroviral therapy is quite effective, and all of these patients are on very rigorous therapeutic protocols, but it is really kind of fascinating. And so we're asking questions like, do you get genetic recombination? What is the implication of having virus in a donated kidney? So far, the early studies have shown that in the first year or two, particularly in the South African study, these patients do quite well. And so we're looking at it from a viral genetic study. I don't think it's going to have major implications. I want to get that out there because I think this is a good thing that we're doing these transplants. But again, a unique opportunity to ask some really fundamental questions. So These are kind of the summary statements from many, many years of research. I can say being in academic medicine has been for me a regretless decision, and I hope all of you are inspired to follow these careers. And with that, obviously, all work requires a team. I've been part of a team in the pathogenesis of HIV for over 20 years. We're on our 5th round of funding. It has been just a remarkable collaboration. We've kind of all spread out across the country, but you can do it from afar. And then I have a great team at Duke that we're looking at the GU tract as a reservoir. So thank you, and I'll be glad to answer questions.